Analytical
validation, verification of test methods is performed in the lab. 1. The lab implements policies and procedures on instruments, test method verification, validation including a. Elements required for validation, verification, as applicable and as statistically significant. B. Documentation of data collection. C. Acceptance criteria. D. Approval of results. 2. A defined sufficient number of characterized samples is used to establish analytical validation, verification. 3. Validation, verification results are documented and signed by the laboratory head, or designee prior to the use in client testing. 4. Actions are taken and documented when validation, verification results fall outside the accepted limits. 5. Validation, verification records are kept for review until the instrument, test is retired. 6. Relevant staff members are trained on how to perform methods validation, verification procedures. Accuracy validation, verification quantitative tests, a minimum of 20 samples with analyzes concentrations distributed across the analytical measurement range should be used. Proportionate mixtures of samples may be used to supplement the study population. Qualitative tests. A minimum of 20 samples, including positive, negative, and low positive samples with concentrations near the lower level of detection should be used. Equivocal samples should not be used, 80% consensus. Within, run precision, repeatability, this precision is performed within the same run. Select a sample or control with enough quantity that has a value close to one of the values reported in kit insert or other references as approved from the lab director, for example. Control reference sheet, biological variation table, and repeat it 20 times within the same run on the same day. Between, run precision, intermediate precision, this precision is performed daily or between days on the same kit, reagent. Select a sample or control that has a value close to one of the values reported in kit insert or other references as approved from the lab head, for example. Control reference sheet, biological variation table. And run it once per day. Note, if the lab uses fewer samples, the lab director must record the criteria used to determine the appropriateness of the sample size. In many cases, a verification study with more samples is desirable.